All right, home stretch. So, how is that blood moving once we get down to the capillary level? Okay, this is the point where we start to get exchanges. Those cells need some oxygen, they need to give back their carbon dioxide. Those cells need some nutrients, those cells need some hormones, those cells need everything that they need to stay alive, whatever their job is that makes them function. So, at the capillary level, where this kind of stuff is happening, that's called tissue perfusion. And maybe you've seen, maybe somebody's had a rubber band around their finger too long and you can tell it's all white and everything. Blood's not getting down there. That tissue is not getting perfused. Okay. When we exercise, we actually increase perfusion to certain areas more. And I've already alluded to this in the past couple of PowerPoints. Look at the difference on this skeletal muscle, 10 times more when you're exercising, increasing the amount of blood flow to the skeletal muscles. And of course the heart, okay, we need that heart to get as much oxygen as it can so that it keeps beating right. The brain stays about the same. Skin's going to perfuse. Why? We need sweat. We need to make sure that we're getting those vessels to the surface, releasing some of that heat so that we don't overheat. So anyway, when we exercise, that however that blood distributes changes. Okay, so down again at the exchange level, how do those molecules get to where they're moving to? Well, it's usually going to be by diffusion, moving from an area of high concentration to low concentration. But going back to that slide that we had those different kind of capillaries, if you remember those. So we had the continuous capillaries, fenestrated capillaries, and the sinusoidal capillaries. Some of them actually go through the endothelial membranes. Just easy peasy. If it's lipid based, then it can get through the membranes just fine. They can get through those hydrophobic tails. Sometimes they're going to pass through those intracellular clefts. So the bigger the cleft, remember, the more things can get easy in and out of. Sometimes they're going to go through that Swiss cheese holes, whether they're small or large Swiss cheese holes. And then sometimes, especially if they're really large, we're going to need to use, instead of diffusion, we're going to have to use active transport. So we're going to have some vesicles that are going to be created in endocytose, exocytose, I guess I should say exocytose, and then endocytose in. So just a reminder, there's a fenestrated capillary, so we've got some simple squamous with their basement membrane. This is fenestrated because he's got the tiny little holes right here. We do have some little tight junctions where there's a little bit of overlapping. Here's a little different way of looking at it. So diffusion. So fatty things can just go right through those cell membranes, no problem. This guy especially, well, the water soluble things are going to use these other methods. So either just sneak right through that cleft right there, or maybe go through those little Swiss cheese holes. And then finally, if it's really big, maybe some sort of protein or something, it's going to get bundled up into a vesicle in exocytose and endocytose. Here's a really important concept about bulk flow. You know, we keep telling you that the vessels are going to lose some of the fluid. Vessels always lose a little bit of fluid. And here's why. That blood pressure, also called hydrostatic pressure, is going to pound against the wall of the capillary. When he's pounding against the wall of the capillary, that's called hydrostatic pressure. The colloid pressure is the pressure that we get from the sucking action from albumin. So your hydrostatic pressure is usually just a little bit higher than the colloid, the sucking pressure from the plasma inside, the albumin inside. So we get a little bit of vessels leaving, a little bit of fluid leaving, and then sometimes it just comes right back in. But anything that is left outside is going to be picked up by lymphatics. That is the term of bulk flow. It's essentially the difference between the hydrostatic pressures and the colloidal pressures. Okay, so the hydrostatic pressure inside the capillary is usually going to be greater at the arterial end, right? Because the arteries are pounding. That's where most of the pressures are. Interstitial fluid, so outside of the actual vessel, between the vessel and the cells, there's hydrostatic pressure there too, but it's really not that big. So we usually would just go ahead and list it as a 0 because it's so minimal. Colloidal pressure. Okay, we got that sucking pressure inside the vessels because of albumin. So it's trying to always keep the water back in the capillary. So the albumin is attracting the water back in. Outside of the vessels, but between the vessel and the cell, again, there's a little bit of osmotic pressure, but it's really not that great. It's only about 1 millimeter of mercury. So that sucking action is pretty small. Who figured out all this? His name was Starling. He was related to Frank Starling, who was in, that was in the heart section. Okay, this is another Starling. This is a different dude. Okay, so in a nutshell, when we're doing the lab, can't remember what it was called, but it's the lab that's pretty much just a worksheet. You're going to have to memorize this. This is almost always one of the questions on the lab test. So she'll give you the numbers of blood hydrostatic pressure, blood osmotic pressure, interstitial fluid osmotic pressure, interstitial fluid hydrostatic pressure. You figure out the difference between those pressures, and then you're going to figure out which direction blood is going to flow. And we'll go through that in lab later. Let's see, we already went through that. You can read pictures. You don't need to read that to you. Okay, so here's an example. So we've got a capillary hydrostatic pressure of 35. And then we have an osmotic pressure of the capillary, so sucking in fluid, of 26. From the tissues out here, we have a hydrostatic pressure from the tissues pounding on the vessels of 0, because it's so minimum, we just call it 0, and an osmotic pressure of 1. So the net, when you plug it into this equation, the net's going to end up being 10 of an outward pressure, which is pretty easy, right? So if we've got 35 pounding on the capillary wall, 26 pushing in, we could even do it in a different way. We could say 35 minus 26 is going to be 9. So we got 9 pounding on this. On this, we've got 0 pushing, and we've got 1 sucking, so it's a -1. So if you bring the 1 to this side, that's going to end up being 9 plus 1. If you remember your algebra, that's 10. But I have a formula that's actually better than this inside the lab textbook, so you will have to memorize that formula and be able to do that. This is just another example showing at the venous end. Arterial end is definitely going to be greater, like I said, because that's where most of our pounding pressures are, really, because veins, now they're fighting gravity, and we're further away from the heart, from the pressures of the heart. You've actually passed all the way through the system, and now it's working its way, fighting to get back to the heart. So those pressures in a vein are really low. That's why when you cut yourself and it just trickles out, you know you just got a vein. And then if it's spurting everywhere, you hit an artery. Okay, so what happens if you end up getting way too high of a hydrostatic pressure, for example? So you're losing too much because of a pounding pressure, or more commonly, when you don't have a high enough osmotic pressure, like when your albumin gets low. Remember, I keep telling you about albumin. It is such an important molecule. Albumin is the guy that's keeping all your fluids inside your vessels. Well, if you don't have a high enough sucking pressure, or if you have too high of a pounding pressure, you're going to have excess fluid leak out, and that's called edema. It can be for many different reasons. I should move over to a picture right here. So if you push on a swelling and you keep a thumbprint, that's fluid that's actually in the tissue. So all that fluid is not in any cells. It's in the spaces between the cells, so interstitial fluid. Oh, I don't have to do this. We've already done this. Like, you're all tired of this. We're all tired of this. Let's move on. I think this is actually the last important slide. I've mentioned this in the lab. This isn't written anywhere except in this one part in the book, and it's actually it's really important. It's also kind of common sense. When you look down at your arms or your hands or whatever, you can see your veins. They're sticking up a little bit. They're very superficial. Arteries are deep. So you do have some veins deep as well, of course, but those arteries are protected. They're closer to the core of your body, and it would take a pretty bad boo-boo to actually make that gusher happen. So most of the time when you cut yourself, you don't gush. If you're gushing, you probably go to the hospital because it's a pretty bad wound. This actually I already mentioned. Remember, you actually have a lot more venous pathways, and a lot of them are interconnected by that term that's called anastomoses. You do have some arterial extra pathways that are connected, but you have a lot more venous pathways. And again, your venous pathways can vary greatly. Arterial pathways, everybody's pretty much got a general same arterial pathway with the rare difference. But venous pathways actually vary a lot between people. You have general areas, and it might be maybe, let's say, if you've got a vessel in your arm that people usually draw blood from, and yours might be a whole lot more lateral compared to your brother or your sisters or your best friends. And then we've actually done those little sinuses already. So this is from AMP1. Maybe you remember those dural sinuses. Those guys are going to be part of the where your cerebral spinal fluid is going to dump back into your venous system. That's where that dumps back in. And then you already know about the hepatic portal system. That's a really weird system where you're actually taking venous blood and you're giving it to your liver before it ever returns back to the heart. So your liver is 60% of the blood that your liver gets is already partially deoxygenated blood. But the reason it does that is so you can get your nutrients from your digestive system into the liver, and the liver is going to process it and decide where all those nutrients need to go. And I'm going to stop talking here. I am going to leave these next slides in here because they are cool. It's got all kinds of vessels in it, but remember, you just need to know the vessels in your lab manual.